Okay, why don't we get started? And then I think there are several important people that are here already, so we should be able to discuss the figure. Can you hear me?
Yes.
Yes,
Okay,
yes,
great.
we have yours. Yeah.
Okay, so good morning, good afternoon, everyone. The agenda for today is first to briefly go through figure two. I think we iterated on this a couple of times, so ideally this is a small, quick thing. And then with the team, we prepared a detailed Google Sheet that facilitate transparency on all the experiments we are doing and should be helpful for any analysis moving forward. And so we were hoping to discuss that to make sure that it's complete and clear. And then and I'm hoping we have a discussion on share analysis after that. And then I have a really quick announcement at the end just to use this platform to share things that the OpenScope project is doing. Okay. As a reminder, we are recording this meeting and I'm planning to post this on YouTube. So please, if something occur that you want us to edit out, please reach out and we can do so before we publish it. Okay. So first agenda is this figure that is here. Can you see it?
Yes.
Yeah.
Okay. So this is following a few iteration. Lucas made this modification based on the feedback we had last week, and then Benedict went through and made further refinement. I don't know if we need to go into details, but it highlight the hierarchical and non-hierarchical version, as well as in the figure at the bottom left, where we had a lot of discussion on dendritic models, was also modified. The OTAPs was removed. In-between neurons was added at the top for feed-forward inhibition. The colors were changed, so there's a sign error and negative and positive error for those instantiation of the microcircuit. Um, I think it contains what people suggested it should, but I want to make sure that they are to make sure that it's fine, and then you all agree on it. Um, are there any concerns, feedback uh, to this change?
I think it looks great. Reflects quite a bit of the things we talked about.
Thank you, Karim.
I have a minor concern. So if you look at the diagrams on the upper side, if you would read those as laminar microcircuits, they would not make a lot of sense. So I wonder if that's an issue or we should clarify that.
How
So
would
particularly
you would
in particular, what I'm concerned about is where we have feedback and top down, it doesn't match the known anatomy in terms of like upper to lower layers and lower to. So I read this not as a top to bottom column, cortical column, correct?
I think there's no assumption. I mean, the error is on the top, but there are no. I mean, that's also why the arrows are not inhibitory or excitatory, right? It's meant to be a higher Maurer level, one would say.
Yeah, maybe we should just have a line in there that says that this is not meant to be a cortical column, or this what's up or below, top to bottom shouldn't be seen as laminar or something like that, just to
Yeah.
clarify.
Do you think that belongs in the caption? Would you agree, or you prefer to have it in the figure, Alex?
I think caption is fine. I think there's just a tendency to look at that and then be confused. Why would there be feedback from the lower to the upper layers if we know it's the other way around?
That's right. Martin, are you fine with this version? Oh, sorry. Karim, you had your voice, your hand raised.
No, yeah, I had just had a question regarding unrelated to Alex. The non-hierarchical version in B, why does it specify visual and auditory?
Oh. Yeah. I think it's an example that was used in other papers. We could remove it. Sensory area 1, sensory area 2. Is that what you meant?
Yeah,
Or-
yeah, because it means that it's exclusive to these two sensory areas, which may not be the case, likely.
I think that's correct. I think it's meant to say that they can be at the same level. Yeah, I agree with, yeah,
Area 1, area 2, or something like this.
that's right.
Just call them area 1 and 2.
I think that's right. Any other feedback?
I mean, you could still rotate the entire figure, right? So just 90 degrees
Of course.
counterclockwise,
Right.
and
Yeah.
then the layers don't become confusing anymore, right? So we just
I mean, I'm biased towards proprioception, so I would, breaks my heart that I don't see proprioceptive, loops part of the predictions because we know it exists and guides all motor functions. So
That's right.
I think the caption would be fine for the first figure, but I do think it kind of makes sense, right? I mean, the prediction error goes from superficial to well, the deeper layer, the feedback comes back up. So isn't it somewhat in the right direction, Alex?
Yeah, I was
I
more
mean-
concerned about A.
In A, yeah, but it's sort of prediction comes from a deep layer and goes up.
Okay.
It's not entirely wrong, I think. Right? I think if it says in the caption that it's not an actual laminar circuit, but because C is sort of more consistent, right? So top down comes the upper layer and feed forward comes the lower layer. So
Okay, so making sure we have the right caption, labeling areas, less specifically on B. I'm not hearing any other concerns, right?
I think it's great. I think Lucas, the others made a very nice figure.
I do think adding the sign and the positive and negative is going to be helpful to a lot of people because I think that point is confusing in the literature. So, okay. So, I will reach out with many of you to sort of put together the caption and the edit to the text, and I think we should be done. Okay, thanks everyone for this work. I think it was needed, but it took a bit of discussion. All right. So the second item is related to all of the ongoing data and metadata at the Institute. Several of you have started doing analysis on our data in the last 6 months, and there are a lot of ongoing experiments at the Institute. And so we took the effort to consolidate all of our nodes. Previously, I think the vision was to share these on the website in dedicated page. So that's why we created this experiment list here with all of the individual experiments, and then you will navigate there. And we also had this experiment summary, but this proved insufficient in several ways. First, as we collect experiment, we need something that is very dynamic for us to add new informations. And so we typically use lots of Excel sheet shared across the teams at the Institute. So we decided to use Google Sheet because it's, very, very easy to share. And so we did the same process of we took all of the sheets across the teams and we consolidated all of them on one. And I'm hoping to walk you through this sheet so that you understand how it's constructed, because it's really important for any analysis. So the first is here, and so all of you have access. You also have edit access, so be careful with what you change. I mean, all of the change are tracked, but I for transparency, I just share it broadly with edit access. So at the bottom left, there's a mice table. And this is all of the mice that we have used so far, including a few mice that we are currently planning to use very shortly. So the first column is the modality. So this all mice are not shared across the modality. There are three modalities: SLAP2, EFIS, and Mesoscope. So these are essentially dedicated mice for each recording modality. Then the second column is whether the mice was considered viable for recordings. There are mice that are not on this table that we never use or never recorded from. I think that's the wrong term. And so far we had decided not to put them in the table because it's irrelevant information to you guys. But just so you know that we are actually performing surgery on more mice that are on this table. The gender, and then the translating details. So you see that this is the two genes. So these are wild types mice. There are one singleton experiments on the VIP mouse, but most of these are either SSTs that express that are used for optotagging of the SST cells in the electrophysiological experiments, or these pan-neuronal calcium indicator mice that we use. So you should have like through this table, you have the detail of exactly what was used for the experiments. And the next column is the virus. So Made had this information very kindly, and so it shows you the exact virus that was injected and the titer of that virus. So that should allow you to sort of understand exactly what was the image for the SLAP2 experiment where we use viruses. Then I have two columns, the birth date and the surgery date. Some of these mice have several surgeries for like doing head fixation and implant addition. So we are going to keep this table up to date as we do experiments with more mice, but this is the start. So the most important information in this table is the mouse ID, and then that's helpful when you go to the session table. That is the next one. So the session table is all of the ongoing neurophysiology recording sessions that any mice go through. And there are more head fixation session on the cluster. So currently, they're not on this table, but so far we focus our attentions on the session that generate neuronal data. So you should see there's a QC field here. This is empty because we envision that in the future we'll take a really close look at these. As the behavior started, so this is when the mouse was started to be introduced to the stimuli, and then the experimental date. So that's the most important column, would tell you on which day boost recordings were done. You can already see that we have more than 59 recording sessions done for this project. So the data is already consequential. The operator is this column, so the self-explanatory, and the rig ID. In some cases, we have several rigs for a single modality, so that should be visible here. So for instance, the mesoscope has 2 rigs, and so we have a single instance of the second mesoscope doing experiments here. The old session ID is, we had an older system at the Institute to track experiments. So for us, it's helpful to have this here. But I think we are mostly using the new session ID now. And it contains the animal ID followed by the date, a unique identifier of the date. So it constructs a unique identifier of that session that we use to track assets as we process it. So this row is really important. It's meant not to change. The mouse ID, you saw it. So you see that the mouse ID is constructed in the asset name. The following columns are the training regimen. So here, those are SLAP2 mice that we did pilot on, and so they were not trained on a specific stimulus, so it's written as not applicable. But down there are all of the cohorts that we use. So for the sake of your interest, I think what's important is to see the motor and the sequence cohort. So you see that we have already started the 2 cohorts for some modalities. And then there is the session stimulus here. So this is perhaps one of the most important information to understand a session. You should recognize here are iterations for the SLAP2 experiments where we attempted several designs. So I went through and added boost information here. And then there is an identifier here that tells you, and this is the name that we use internally to track session. And so for your sake, what's important is the last beats. So sequence, standard, durations, or sensory motor are the four design that we use. I have some things in red here. This is meant for ourselves to double check because as we did this consolidation across tables, there were some bits of information that were either inconsistent or something that we need to act upon. So this is where we'll likely change the value. But you can see that there are mice that I went through all of the 4 session design in several modalities. Okay, so this column is mostly empty. I might fill it in later on. It contains the path to the exact code that was run on that session. So if you go there, you get on GitHub and you see the bonsai workflow that was used. I want to make sure the implementation is accurate, so I haven't had the time to do that. Then the next column is the workflow state. I think currently it can be either planned or acquired, so or processed. So I should edit this, but this is helpful for us to know when an experiment is finished, and then Carter or others can trigger the processing and pushing it up on CodeOcean. So this is going to be helpful downstream, but you can see which session was acquired. There are sessions that are written as aborted. So I thought it was helpful to show them. In some cases, we decided midway through the experiments to end the session. That could be for technical issues or for making sure the mice is comfortable during the experiments. We don't want to push beyond there. And so you should see these instances here several times. there are notes that were added in the last week. I think some of these could grow. I consolidated some notes across multiple sources. And so this is helpful context to know what happened during the session. So for instance, in this case, the SLAP2 experiment was started. We noticed some technical issues with motion corrections because we have online motion correction in the SLAP2 rigs. And so we decided to abort it 1/3 of the way in. And so the mouse typically will be re-recorded the next day or so. And so these are to give you context of why and what happened during a session. Okay. The next few columns, so all of this, the column in green are meant to be shared across all modalities. So these are like general status of an experiment, and they are filled in by the team at the institute. You shouldn't have to touch them. The next in yellow are, because as you know, we are using CodeOcean now to push our data to the cloud. And the first column here just say the status. So this is helpful for us to know which session we pushed. So you might see that we have a lot of no's here, and this is because that process is ongoing. Once the session is on CodeOcean, there are several assets. The first one is the raw data. Then we have a process asset. So for instance, in the case of spike sorting, this is running kilosort. So we have an automated pipeline to do this. And so that generates a second assets that we need to track. And then once we have these, and there are many, many steps into the pipeline, we also generate an NWB assets on CodeOcean that is the piece that we upload to Dandy. So our intent is for all of those assets that you see here to put them in the public collections on CodeOcean that you will all have access to. So that means you'll have access to the raw, the process, the NWB derived assets through the public collection on CodeOcean. There's already a public collections, but we added the EFIS assets. But in the next week or so, we'll do the effort to add all of those that are listed here to the public asset. So you will have a sort of full access. But you see that we still have work to do to sort of finish all of those uploads. The next column is in orange. And so this is the data that you have access. So again, similarly, wherever it's on Dandy, and then this is the path to where you can find the single session assets. And so we've shared a smaller subset of all we have collected so far. The following columns are modality-specific information. And so far, we don't have any on the behavior cluster. We might add some in the future. EFIS as a common coordinate framework, a registration asset. So this is a step where we take the 3D volumes of the mouse, acquire imaging data to register the probe to the common coordinate framework and register these really carefully, so that every unit is within a specific area that we calibrate. So we have that, and that process takes weeks or months, depending on the availability of the imaging pipeline and the time it takes to do this process. So some of these have it, some do not. And so this is, for instance, a case where it will be helpful for you to see when it becomes available, so that you can have a use of it later on. But it's likely that we will push EFIS recording earlier than they are registered to CCF. Okay, I'll take questions after these columns. Then the last one is something that several of you made request, and because SLAP2 is a more recent platform, and we had less consolidated process to sort of track all of those experiments. And so we adding a little more information here. So the first column is the intended channel, recording channel. So it shows you the similar information as what was on mouse, viral information, but condensed in a way that is a little easier to use. The two last columns at the depth. Most of the time we had that info, we realized that in some experiment we might not have recorded this. So I put question mark in some of those cases. We'll double check if we can extract the exact micron location. Here I know we don't have it, but I wrote exactly the assign compartments so that it's helpful for you. There's some cases where it's aborted, so you won't have it, but you see that we have a fair amount of SLAP2 data already. Yeah, so that's the content of this session table. Any questions on this?
I have one. So when I take a look at this spreadsheet, I don't have a good feel for the amount of work that will need to be done taking any of these pre-processed data to get to the main, let's say, the main scientific question we're trying to address in a particular experiment.
Yeah.
I know this is how to find the data, but to get to it, what to expect in terms of the type of pre-processing that remains to be done or verified before we can jump into the real thing.
That's right. So the answer might depend on the modality. So there are 3 modalities, right? We have ePhys, Mesoscope, and SLAP2. So I would say the ePhys, we run kilosort, we have metrics on those. We have a QC portal that we can give you access to, so you will see the quality of the metrics that I extracted. I think it might depend on your questions, but like for instance, if your question is very sensitive to stability of units across the entire session, then you might have to be carefully checking those units. We typically start the session with a 30-minute delay to reach a regime where the recordings is very stable throughout the entire session. But I think it's really important to be aware of any potential caveat in the analysis. And so for ePhys, you should expect that the data has been processed. There are units that are extracted. You could use it, but it's really important that any analysis is done carefully. For Mesoscope, is a similar situation. So we have a pipeline that runs. It's going to extract ROI with traces. There is neuropil corrections and so on and so forth that are run. But depending on the particular analysis that you care about, the exact pipeline could influence your results. And also if your result is very sensitive. So that's why we in both ePhys and Mesoscope, we also share the capsules that show the code. In fact, those are public already, but I think we will try to put them in a public collection. Right, and we will package within NWB. So I expect that any analysis is like any other analysis you do in your lab. You have to carefully characterize the responses and make sure that there's no artifacts that confound your results. SLAP2 is a more junior pipeline. I think the processing in place is very good, and some of you have played with it already. But it's probably going to be a little more collaborative with the SLAP2 teams. I anticipate that it's important. Yeah, does that answer your questions, or Karim, or because all of these modalities are processed. So in principle,
right. Yeah. So the reason I mentioned this is because we started analyzing some of the SLAP2 data a while back and got to the point where we wanted, let's say, for example, the coordinates of the synapses
That's right.
that are being recorded and so on. So it was kind of, hit a roadblock, and then I lost track. Maybe Nicholas would know a little bit better, but we just need to be ready because once we get into it, we want to just, be dedicated to continue to analyze a specific data set without being interrupted and then having to steer directions and get back to it later on. So, just the traction,
That's right.
Gaining traction in something would make it more productive.
That's right. I do appreciate the need for support for one specific modality or others. It's just as you can see, we're just requiring data all on three fronts, and we're not there's sort of a gigantic team here, so we have to sort of dedicate attention to certain things. And so this table is to sort of try to start to address some of the concern that yeah, Nicholas raised and others about access to the data and metadata. And I think what for us our next step is to make sure that we catching up in upload so that you have access to all of those sessions. And then once we are done with all the experiment, I just anticipate we're going to have another round of cleanup, making sure, because we essentially want to generate a large set of sessions that are well characterized and described, so for all of you to analyze. But yeah, that's going to take a bit of iteration.
Jerome, can I add something, please?
Please.
So for the e-phase, I wanted just to make it clear. So after the data collection, there is the QC step. So that can take a little while to review all the units characteristics and everything from the spike sorting. But then the thing that takes the most time after that is like there's 3 weeks of brain clearing. So we have to wait 3 weeks before the brain can go to imaging. And then once they are in imaging, sometimes we don't know when they will be actually, because there's a problem sometimes with availability of the microscope. So we don't even know really exactly when they will be imaged. So please be patient with us. We're trying to keep the pace and everything, but it's also very important that everything is well done and everything is checked thoroughly. So that's what I wanted to say. Yeah.
Agreed. Any other questions on those tables?
Jerome, just rule of thumb, when do you think, roughly speaking, the data collection, or at least for one modality, the data collection might be done? Because that, just like Kareem said, it might make sense for us at that point to consolidate some of that to in other ways. So for example, I can imagine I'd be happy to do that to, for example, visually read out the information in the Google Sheet and create some visual graphics in terms of, if you're interested in this paradigm, you have this many sessions in this modality and that modality, and here are the files, just to organize it a little bit more high level and simple for somebody who might just want to get to quickly. But it makes most sense to do that once the data collection is done. Ideally, once the upload is done, but just so we have a timeline.
Yeah. I mean, all three modalities are going full steam. And so I think I anticipate that late spring will be done. I don't want to put a precise date because we have to address like quality issues. We might, like the exact amount of data that will be collected is up to change. But we really want this to be done late spring. And and so that will mean this table will be completely full with all of the assets on CodeOcean probably around that time range. I mean, That's a lot of work. I mean, we're talking about probably on the order of 100 session to 200 session. Yeah,
Can I ask also,
maybe.
there are some mice where we managed to take only 2 sessions out of them instead of 4. How usable is it for the community to have these mice, or is it really not usable at all? So what do you guys think about having 2 sessions instead of 4, sometimes when it's impossible to do otherwise?
I think we need to go to this.
You mean with neuropixels, 2 neuropixel
Yes.
sessions?
Yes.
And
Like,
so
let's say that
it
we have 2 sessions that don't pass the QC and 2 sessions that passes the QC. So how is that usable for you guys?
I don't see why it would not be usable, but
Okay.
maybe other people have other opinion. I think even 1 session is usable from my perspective, but
Okay, good to know. Okay.
So, Severin, with respect to the QC, I mean,
Yeah.
depends. Some quality is lost in certain aspects, but it might be, adding to the number of animals for a particular question. That's definitely a good thing, even though the data is incomplete.
That's a special case of stress actually. So the eye is closed. So basically there's no visual data whatsoever.
Oh, I see. Okay.
Mm.
And I think what would be helpful, Alex, regarding your points, is as we are collecting data. So this is what Severin is mentioning here. We are working on 4 sessions for NeoPixels, right? With each a different context, and so there are cases where one of those, 2 of those, or 3 of those context cannot be recorded in a mouse, and so that means uh, some questions that rely on comparing uh, responses across context in the same mouse will be difficult to answer with those sessions. And previously, I mean, Karim and many others put together this figure here that highlight different key analysis goals, that we wanted to do on those data assets. And so I think what will be helpful, maybe, that we get started on is how each analysis goal depend on, uh, specific needs for the mice number, so that we make sure that we acquire the data that allow this analysis to be done. And um, yeah, so in as you said, Alex, some analysis wouldn't be insensitive to issues like this. Some of us might, and for us to sort of guide the data acquisition, I think that might be helpful to have that table that you were thinking of, Alex. Yeah.
yeah,
Let's see
my
if I
main
can-
idea is my main idea. So I love how thorough and rigorous you are in collecting all of this information, but I can also see as new people might be entering the project that this is overwhelming. And so I think that should do both, exactly as you said. Like first we collect everything, and then I can see a second step where we condense things down to somebody who's just, look, I want to analyze this and this. Why do I need to know uh the ID of this animal or something, right? And so like we can condense it down. It's like, well, here's the data, here's the code, and get going. And so that's what I envisioned, that we have another interface that simplifies things for somebody who they can look up the details later. We make sure that they can always find the entire information. But if they just want to analyze one paradigm or one modality, that they can get started quickly.
That's right. But I think my question was different. Is I think it will be helpful to us if some of you go through the analysis plan that we wrote in this piece and highlight how the data that is in this table currently or will be will be used for different analysis goals. It could be high level, but and then maybe make recommendations on how we should address QC issues for each analysis, and or even say if we don't have these mice, this analysis is compromised, and so then maybe because we don't have infinite resources, and so where we dedicate it could be a decision point. Like okay, this analysis is really important, or and I think I need your leads on Boost feedback.
Okay, so I have a comment. So typically, to understand, for example, how many animals we need for a particular type of analysis, let's say sensory motor mismatch,
That's right.
we start first by knowing how much variability exists in the data. So,
That's right.
it could be that the standard mismatch where the mouse is not doing any motor tasks, right, is less variable, compared to the session 2. And so maybe the number of animals in one session or one context would be different. And so I guess my answer to you is without having a good grasp on how much variability exists within each of these contexts, it's hard to tell whether we have enough animals or enough sessions for a particular context or not, unless you have that number already predetermined.
Well, I mean, we did some work on power, but we already have a fair amount of data to help guide this.
Is that in the manuscript?
No. Well, yeah, in the manuscript there's a part about statistical analysis in the end.
Right.
But I'm also saying that now we have a fair amount of data. Like we have some SLAP data, we have some NeuroPixel data, we're going to push soon some classroom imaging data, and so I think
So
I think
is
you
the
might be
question-
empowered. You might be all empowered soon to sort of start digging into that questions, like how many experiments are needed for the different questions.
Okay, so is your question how much more sessions to allocate to each modality?
For instance,
For instance, so if we already have a good idea about how many we would need, given the statistics that we ran going into this manuscript, so I'm not sure I understand what the guidance we can provide here.
well, because the failure mode, the challenge we face are multi-dimensional. Like I don't think we've done predictions like, okay, if you have half of the session for NeuroPixels, how does it impact that questions? What, like the goal, the target could be specific to each analysis. I feel like maybe I'm wrong, but I feel like it would be helpful to have a first attention to to the different analysis goals, and then provide to us some inform. Okay, so these questions really need the 10 mice. This question can live with fewer mice. It's like so some guidance there.
I see. So I can tell you just, at the top of my head that any analysis involving information, calculation, mutual information, bit rates, stuff like that, does require a lot more data than pretty much anything else, because you're working with probability densities that are not even close to the true ones. And
That's
so,
right.
so yeah, I mean, it definitely depends on the type of analysis, and therefore we wouldn't be able to answer this question precisely unless we start really to dig into the data and see how the analysis varies from animal to another, from session to another, from modality to another. All these are still
Yeah.
unknowns.
I don't know if that's true because we did share NeuroPixel data already.
Well, we didn't work with these data yet.
No, I'm not. No, I know. I'm
Yeah,
not.
yeah.
I'm not pointing at you, but
talking about
I'm just
myself.
saying
Yeah.
that. I'm not just saying that we are starting to share data, so I feel like getting a sense of variability will be helpful.
I don't know if any members of the group have already started to analyze the NeuroPixels data. I mean, I don't recall. I haven't been. Okay, so
Yeah.
how is it looking from your side, Alex?
I didn't. I forget. Somebody in.
Farzaneh and
Yeah,
Ali did.
Fasani's lab looked at mostly the local feed potentials. So yeah, it's a very first pass analysis, right? So immediately we ran into some kind of questions about do we get the layers right and so on. But I mean, we had visual responses. So but yeah, the way that I see it, like implementing something like you're showing here, right here, Jerome, is months of work. So I think and so that immediately will stop anybody attempting anything. I think we have to think about baby steps. Like we have to, like what we did with the slab in terms of, okay, can we look at the receptive fields first? Can we look at the orientation tuning first, and so on? I think that will help. Like we should identify what we need and go step by step. For example, do we have visual responses across all areas? What are the latencies and so on? To build up to this level of complexity. It's going to take a while. But the good news is that once you have some code, it's easy to build up on that. I think that was our experience doing the SLAP experiment. So it's great. Somebody writes code how to read in the NWD data. Somebody wrote code how to find the different blocks. So we now know which block is block. And each step along the way, we ran into small issues. Then once they're solved, somebody else can take that and look at something else. So I think my impression is that is what might happen here as well, organically. So I think what we can't do is write a data analysis pipeline, keep collecting the data, and then just run it through that. I think that might not work. It's too much work on the analysis side.
See. So what would be your preference, Alex? I mean, to organize this effort, because
yeah, I think that we should have. I like the idea of quality control, but on different levels. So we basically, what we did in step 2 was a certain way of quality control, right? We said, hey, there should be receptive fields and so on. And so we did find problems this way in terms of we didn't even know certain blocks weren't labeled in the data and so on. And so I think we should do the same for the NeuroPixels data and then as well for the Mesoscope data. So baby steps. Do we get receptive fields? Do we get visual responses? Do they make sense? Sanity checks along the way. And so that's not gratifying. I understand it would be nice to immediately look at prediction errors, but there's just too much room for error that, it's grunt work, but it's the kind of grunt work that's needed between data collection and high-level analysis. There's just like, usually that's 80% of the work. There's just so much in between of making sure we have the data exactly as we think we have and so on. And that can give you already some feedback in terms of if the visual responses don't make sense, or because these are areas that are new to NeuroPixels recordings that, there's not a whole lot of responding neurons and so on. We can then adjust the statistics and say, look, if we want to find a prediction error, but most of the neurons don't respond anyways, then we understand we need more data, or vice versa.
I see. I think that makes sense. I think what I'm calling for is making sure that the data we collect is being looked at as it's being collected, because there are important feedback that can influence what we do. And so I fear the situation. Yeah.
I agree with you. And so, so what I'm wondering is if we can incentivize some of that. So for example, I won't be able to do anything until May because I'm totally swamped with teaching.
Yeah.
So it would be only in the semester break that I get time to code. Now, I suspect with the NIH funding situation, NSF funding situation, what it is, other people find themselves in that position as well, that we can't get as much course buyout as we used to. And so a lot of us are just more busy administratively and in terms of teaching. So it's the best of breaks where we can actually do research. So that from my side, it would have to wait. But so for example, we could incentivize, here we have 3 modalities now, SLAP and NeuroPixels and Mesoscope. And again, do something simple, like just look at the receptive fields. And I think what's interesting is that, yes, ideally we want to finish this giant project and have this giant paper. But because SLAP is a new technique, we might, just by looking at receptive fields, get a very interesting paper out of that. So there's an element of discovery science here that might make it interesting, even though it sounds like that's just infrastructure kind of work. that just by comparing what we see in terms of receptor fields across 3 modalities, that's new. Nobody's ever done it in the history of the universe. So, but then at least we know we have a very concrete goal. We want to see the receptor fields across 3 modalities. That will allow us to make sure we got the trial triggering right. We know how to load the data across all of these modalities. So a lot of the stuff that has to be done anyways now will get done, but we're not shooting as big as saying, okay, we should do all of these analyses as they are here with all of the different data, because that's a lot of work.
I understand that the bandwidth to do analysis is important to have, but would you be collectively open to sort of work with us to sort of flesh out those baby steps? Like, because I think we wrote a comprehensive analysis plan, and in this plan there might be some of those baby steps that we want to sort of prioritize. And I think if we make this plan clear, it will be easier for incoming students to take those and then push it, because there are many folks that reach out is like, how can I help? Like if we don't sort of advertise a clear plan on how they can help, it's hard to sort of.
Yes.
And
So we can even set up a second meeting where some of us can help mentor and advise. But yeah, as I said, so for, like my experience with the SLAP2 data has been that in trying to analyze the data, we realize it's not even as trivial as like, well, here's the data, right? So like, how do I download Neuro Data Without Borders, NWB data? Okay, now I got the NWB data, but what are the blocks? So we realized, oh, that the blocks aren't labeled. Well, let's write an algorithm that detects which block is which. And so so that's I think stuff that we're probably, and we don't even know what's going to await us until we do it, that we realize, oh, there's missing information, or we need to do this here, or we, and then I think we also have different expertise. I think the interesting part of this project is some of us have worked with 2-photon data, and we're, probably very good with photo bleaching and everything that comes with it. Some of us have worked with neuropixels data, and, understand what current source density is and everything comes with it. But it's going to be a challenge for each of us to learn what the other people are doing and how the analysis works. So that's why I'm saying, like, just by saying, can we do receptive fields across these techniques, we should encounter a lot of these issues. And we might get something that's already publishable and interesting, since these kind of comparison across techniques haven't been done before.
Okay. So how would you approach this? I mean, would you have the time to review the analysis plan we wrote and extract collectively the baby steps, or how would you? Because it's a bit of effort. I mean.
Yeah, I can. As I said, I'll have more time starting in May. Like, but what I would do again, how I would, like, we're facing a monster of data and complexity, and so what I would do is again focus on receptive fields and orientation tuning, and make sure that we have several sessions in different modalities, 2-photon imaging, neuropixels, SLAP. They're all complex. The neuropixels, bunch of areas, bunch of electrodes, and so on. But can we get just receptive fields for all of these 3 different techniques? Or can we get orientation tuning for all of these different techniques? And so once you have that, then at least you know that how to load the data, how to analyze the data, and so I'm not sure we're there yet. And so once we have that, at least we got that step, then we can take other steps. And so likely what's going to happen, what happened in SLAP, is that people all of a sudden ask scientific questions. So there was, all of a sudden we got interested in how receptive fields are distributed among the dendrites of a neuron. So what I expect is as the analysis comes in, we realize, oh yeah, there's actually science that we can do with that. So it sounds trivial. And I also want to do predictive coding right away. But as we slowly build up to that, I think it might help. So just as you did before, let's say we want to have the receptive fields across the techniques and orientation tuning. That should be enough to get us started.
Yeah.
And it's important sanity. It has to be a figure in the paper anyways, right? So if you're saying I got 2-photon data, probably a reviewer would say, well, show me that you got receptive field mapping right. And so we need that anyways.
May I suggest something? Because I have to run in a couple of minutes. So
Yes, please.
now that this manuscript was written maybe a year ago or so, I can't remember, but now that the data has been, the experiments have been designed, the data has been collected, the analysis is still, starting, or maybe started but not full blown. Maybe we can start thinking about the problem in reverse order. Let's assume that we take Alex's suggestion and say, okay, what can come out as far as a paper out of just some preliminary analysis from this data, and then figure out, okay, what will the figures will be, right? And then we can start to designate, this as part of the plan with quantifiable milestones. Okay, once we reach that particular step X, then this is one paper that will be packaged and sent out for review. And then we can define what is the next paper that we want to get out of this. So anybody who's coming into this and trying to help with limited amount of time and limited resources would be able to go and identify a chunk in the analysis and, get to invest their time in it with a clearly defined metric of reward, if I were to use that word. Part of the issue that we identified early on is that when we, the first lab 2 data that kept coming, Nicholas, for example, had allocated quite a bit of time to look into it, and I assume other people did as well. But then we realized at some point that it's taking a lot of time to go through all the things that Alex has mentioned and other things that he didn't mention. That was taking too much time to just go through that and to just get to the point where we can identify, hey, we've identified something in that data that might be interesting for people to weigh in on. And so, we left it there, and then we're trying to slowly come back to it. But then I think the mindset has to change here, is that we have to, shoot for a well-identified milestone in terms of analysis plan and potential paper submission. That's what I wanted to say.
I think that's probably right. But do you think I think what I'm asking is, like, could we spend sometimes within the next week or so reading through, because we wrote analysis goals, and maybe in one of those there's one of those that could be our focus, and then I'm happy to help organize around a focus, but I think I'm also sort of hoping that you could help, like, just because we wrote a fairly consequential analysis plan, and so maybe one of those should be our focus, and then we sort of, as you said, craft a paper, and then and have like a really clear reward goal. But I don't want to sort of choose it for you guys. I mean, we need I will need your input to
Yeah, and I don't think that this is going to be simple to discuss in this group-wise meeting. It has to have some dedicated set of meetings to go through these specific plans. I think so. That's just, but feel free to push back.
So how would you organize it? Like, should we have like a one-by-one focus discussion?
I mean, the people who are involved in a particular data set or set of data or contexts or specific modalities should be in that meeting if they have the bandwidth and the time to help out. So maybe initiate a poll for people to fill out and say, okay, we have this, let's say, sensory motor condition, and we have this type of analysis that we identified would be the most immediate and most important one to go through. There are other pieces of analysis, but we're going to just prioritize one or two. And we need two people to start to take a look at the data and tell us what's happening there. And then people can enter their preferences, and then you can identify a subgroup of people who are, very interested in this problem, and then we can go from there.
Yeah, I second that. I think a poll might be exactly how to solve this. So we can make a poll for each of these analysis goals and see what is most popular, and then get started with that. And these people can form a subgroup to organize themselves.
because without that, everybody's kind of free. Okay, if the data is available, anybody can pick and choose. You might end up with people doing redundant analysis. You might not end up having enough people to do, analysis for a specific context or whatever. So, leaving it out in the open like that for people to pick and choose what they want is, is not very organized. We need to just concentrate on the set of things that are immediate milestones to achieve, and then see who is interested through some polling or something and then organize them into smaller meetings that would just be very focused on what needs to be done for the set of sessions that data was collected for.
Okay. I think we could organize a poll. That might be a good idea.
Yeah, and I agree with Karim. I had to from apart, I had the same experience, I had the same concern that especially some junior people that got quite active in the project got discouraged by that. Despite a lot of effort that went into it, it seemed the progress was very slow. So I think that we can think about exactly as Karim said, like very concretely, here's what's needed for a paper. And so like that's a paper that should be doable in, let's say, 6 months or something like that. And so more senior people probably have a good feeling for what would be needed for what kind of paper, and then use that as a guiding star for where the effort should go into. Because otherwise, I'm also worried that people would join and get burned out and leave the project. So yeah, I like the idea of maybe we can vote what seems the simplest and most attractive, and then even there maybe identify exactly what would be needed for, like, as I often do it with my students too, almost like draw cartoons of what figures would be needed for what paper, so that it's very clear what has to be done.
Would you be helpful? Would you be open to sort of help me shape that poll? Because I feel like if we just need to put a little bit more thoughts into it. What are the right questions? And I agree that could help consolidate intent. Yeah, I mean, like a 5 or 10 questions poll with like, what these are the goals we highlighted in this paper. Where should we focus first on? And then have people vote in which one they want to help with. Okay. And maybe next week we can review the poll and then send it to everyone.
Sorry for interruption. I just wanted to add a
Please.
point here. For the files that we worked on for NWB file, I think we prepared a good, well-designed notebook that Alex also checked that, I think. So people can use it to read different trial types, block types, everything that's designed for each of the different types of sessions. So it would be really, I mean, it is not a cold source for people. They can also jump into the further deep analysis. And also later on, I shared another notebook that I did some pre-processing, like adding a notch filter and these stuff to get rid of some of these noises and these stuff too. So if you have time, you can check it and also use it as a start point for other people to use that.
That's right. This is really helpful, Hadi. I think what we're trying to see is how we can leverage those pieces to specific questions and
Yeah, sure.
help coordinate those so that you and others can make progress towards certain goals.
Sure. Yeah, I just wanted to, I mentioned
This is really helpful. Thank you. Okay. Well, it's 10:07, so maybe let's try to yes, team, Stem.
Hi, Drum. Yes, I'm with Forrest in this lab, and I think originally we were planning to start our experiments to add to the data set sometime this spring. I don't know about the exact timeline for that, although I do have our rig hardware and software built and programmed, although I would need to get with you to make sure, okay, we're using a consistent session structure and how to structure our 2-photon imaging data set so that it aligns with the other modalities.
Okay. It seems like we should have a one-on-one here. Do you agree? Or?
Right, I think that would be helpful because looking through the paper and the websites, still I would need to make sure, okay, when we generate the Excel spreadsheets that define the stimulus session structure, to make sure, okay, we're doing that consistently so that when we get to the data analysis, we have a good comparative look between the other modalities.
Okay. Let's schedule a time to discuss that. I think that's specific to your technical needs.
Right, sounds good.
All right, so I will start a poll and then I'll send it your way, Alex, and maybe Kareem also want to wait on it, and then that might help us consolidate our efforts. Sounds good.
Yep.
Thank you for the input.
Of course. Take care.
Have a good day, everyone.